Alright guys, so we're back for concept 5.3, which is all about the lipids, which are going to be fats and oils. Before we get onto the lipids, let's just do a quick reversal or summary back to where we were talking about carbohydrates. And carbohydrates are the various types of sugars and starches, the first large biomolecule that we were speaking of, macromolecule that we were speaking of. And just to give you another example, you may know or have heard about that some people are lactose intolerant, meaning when they drink milk or they have dairy products, cheese and ice cream, their stomach gets upset or they have a lot of gas or they have problems digesting milk related products. And that's because for many adults, particularly of certain lineages or races or where they come from, they're lactose intolerant. And what that means is their body lacks the enzyme necessary to break down lactose, which is a disaccharide, into glucose and galactose, which are the two monosaccharides that make up the disaccharide lactose. So almost all humans, as infants and children, they produce this enzyme that breaks down the milk sugar called lactose. But as they become adults, they lose that ability, or some of them lose that ability. Now, in modern times, there are many humans who persist with that ability. They can drink milk and process lactose all the way up into adulthood, which is actually a mutation, a couple of different genetic mutations that have resulted in that. So in populations where milk was readily available and was part of the food source for that population, you tend to have a higher percentage of people who can drink milk and use it as a food source all the way into adulthood. And then in other parts of the world where milk maybe wasn't as readily available or was not used so much as a food source, that population tends to not have that enzyme into adulthood. So they have problems digesting milk and the lactose sugar in that milk. So again, that's a good example of one of these polysaccharides, or this is a disaccharide, lactose, which is broken down by the body into two monosaccharides, glucose and galactose. But that requires an enzyme, and some people lose the ability to produce that enzyme as they grow into adults. So you can take medication. There's like pills that you can take called lactate or other sorts of pills, and they contain that enzyme that your body has stopped making. So if you want that ice cream cone and you're lactose intolerant, well, you can take a pill or chew this tablet before you eat your ice cream, and hopefully that will help you so that you won't get sick to your stomach. All right, so those are our sugars and starches. Those are all carbohydrates. We're going to move on to a different group. This is lipids, which are fats and oils. So concept 5.3 says lipids are a diverse group of hydrophobic molecules. We know that word hydrophobic means water-hating or water-fearing, so they don't mix well with water, and you probably already know that when you make salad dressing and you mix the oil and water, or oil and vinegar, they don't stay mixed, they separate from each other. You've seen probably oil ride on the top of water. Like if you go to the Walmart parking lot after a rain, you can see oil from cars sitting on top of the water and shimmering with that iridescent look to it. So hydrophobic means water-hating or water-fearing, and so lipids are a wide variety of molecules that tend to be hydrophobic. Now, lipids are one of these four main categories of biological molecules, but normally we don't put lipids in with those other three as macromolecules because most lipids are not as large and complex as, say, proteins, carbohydrates, or nucleic acids. So they are an important part of biology and chemistry, but they're not usually considered macromolecules like the other three we're going to look at are considered. Lipids are the one class of large biological molecule that don't really form polymers, or certainly they don't form long polymers and chains like the others do. So some of these are repeating patterns, and they're like short chain molecules, but normally we wouldn't call them polymers. They're not long enough and complex enough to be called polymers. The one unifying feature of most lipids is that they have very little or no affinity for water. That means they're hydrophobic, of course, and the reason they're hydrophobic is because the majority of their bonds are between hydrocarbons, which form non-polar covalent bonds. So remember, non-polar means there's no negative and positive sort of side, and so because water does have a negative and positive sort of end, the water is much more likely to be attracted to itself than it is to the lipid. So it tends to avoid the lipid and stick to other water molecules and that's where you see that hydrophobic reaction. The most important lipids that are important for biology are fats, phospholipids, and steroids, and we're going to look briefly at each of those. We're going to start first with the fats. And fats are made from a combination of two smaller types of molecules, something called glycerol and something called a fatty acid. Glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon. And I'm going to show you a picture of this in just a moment to kind of remind us. And a fatty acid is a carboxyl group attached to a long carbon skeleton. And it's fairly long, usually 16 to 18 carbons long. So even though fats are not considered polymers or macromolecules, they are made up of smaller molecules via dehydration reactions, and they do have like a repeating chain pattern. They're just not quite large enough, long enough, complex enough to be considered macromolecules or polymers like the others are. So let's take a quick look at a picture. So here we have the two different parts of the fat molecule. On the left, we have the glycerol portion of the fat molecule, and like we said, that's a 3-carbon chain here with a hydroxyl group attached to each carbon. There's our little hydroxyl group, our OH, so you can see how that's attached to each one of the carbons. And then the other part of the fat is this long fatty acid chain. You've got this sort of backbone of all these carbons, and so it's going to have anywhere from 16 to 18 carbons, and it's got a carboxyl group attached to that skeleton. And our carboxyl group is this right here with the carbon, with the double bond to oxygen, and then the bond to the OH here. So if we bond these two together, that is a dehydration reaction, kind of like what we've already talked about with creating polysaccharides out of the monosaccharides, or polymers out of monomers. So we have this combination that goes on right here, and that produces sort of as an excess or as a offshoot this water molecule. So this is a dehydration reaction that joins this fatty acid chain to the glycerol molecule. We already said that these fats are hydrophobic. They separate from the water because the water molecules are more likely to bond together with hydrogen bonding than they are to the fat. So they tend to ignore the fat and bond with each other. When we're creating these fats, we've talked about this chain. Let's go back to the picture. This fatty acid chain that's connected to a glycerol. Well, actually, you'll notice there's 3 spots where that connection can occur. So that's actually what's going to happen. That glycerol is going to have 3 fatty acid chains connected to each one of these spots. So let's look at a different picture here. We call this a triglycerol or a triglyceride because it has 3 chains, 3 fatty acid chains connected to the glycerol molecule. You'll also notice that not all these chains have to be exactly the same. Like this chain is shorter than the others. So these can be 3 different fatty acids or a combination of 2 different fatty acids, or they can be the same. But there's going to have 3 long tails or fatty acid chains connected here by what's called an ester linkage. This linkage between the glycerol and the fatty acid is called an ester linkage, and you'll have 3 connected to each one of these spots on the glycerol. So you've probably heard in TV, perhaps commercials or shows related to health about triglycerides when they're talking about fats and things of that nature. Well, it's called a triglyceride because it is a glycerol molecule attached to 3 fatty acid chains. Down below here we have an animation. So if you want to right-click and play this animation, you'll see this combination. It's going to be a dehydration reaction that occurs, and these 3 fatty acid chains will connect to the glycerol. Just as a side note, you may notice that if you have grease on your hands, whether if you've been in the kitchen helping cook, or you have oil or grease on your hands, or perhaps you've been out working on your car or working on the lawnmower and you got oil or grease on your hands, it's very difficult to wash off with just water, and that's because of the hydrophobic nature of those oils and fats and grease. So we usually use soap, and the way that soap helps is soap is made up of long chain molecules also that tend to have one polar end and one non-polar end. And so the non-polar end can attach to the oils or fats or grease, and the polar end can attach to water molecules. So the soap kind of acts like a bridge between the water and the oil, or the water and the fat. It connects them together so that then the oils and fats can be washed off of your hands as you rub them together and run them under the water. And also you'll notice that hot water usually helps. That just provides more energy, so the molecules are moving around quite a bit more, knocking into each other more, and so that helps get the oils and fats off of your skin as well. Now, we did say in that triglyceride, the three fatty acids don't have to be all the same length, so they can have different numbers of carbons in them, and also they can differ in where there are double bonds. So in some of these fatty acid chains in various locations, say along the chain, you'll have some double bonding occur as well. You don't in any of these that are shown right here in this picture, but I'll show you another picture where they do show some double bonds. So based on the length of these chains and which chains they are and where these double bonds are located, that tells you what type of fatty acid we're talking about. There's two different categories. One of them is called saturated fatty acids, and one of them is unsaturated. And again, you've probably heard these terms if you've ever watched any health shows, or maybe in health class, or you watch something on TV about cholesterol and fat and diet. You've probably heard this word, saturated and unsaturated fatty acids or fats. Saturated fatty acids have the maximum number of hydrogen atoms possible and no double bonds. So if we go back to this picture, this would be the creation of a saturated fatty acid if we're looking at these three fatty acids in the glycerol. Because notice, every carbon is filled with hydrogens and there are no double bonds between any of the carbons. We would call this a saturated fatty acid. That atom or that molecule is saturated with hydrogen atoms. It can't take any more. Now, an unsaturated means there are one or more double bonds. And when you have double bonds, you're displacing one of those hydrogen atoms. So let's take a look right here. A saturated fat, like butter. Butter is a saturated fat. And if you were to look at the model, or you look at the 3D model here, every one of these carbons is saturated or filled with hydrogens as well. It can't possibly have any more hydrogens. And you'll notice it's kind of in a nice straight long chain. Now if we look at an unsaturated fat, saturated fats tend to be solids like lard or butter, something like that. Unsaturated fats are more of your oils. They tend to be liquid at room temperature. And what you'll see is in one of these chains, right there, we've got a double bond. So right there we have a double bond. And that means there's no space for a hydrogen there because that double bond is taking up one of the spaces or electrons that the hydrogen would take. It also gives it this kind of odd shape, this kink to it. So you see how it's turned or kinked right here. Well, this double bond means that it's unsaturated. It doesn't have the maximum number of hydrogen atoms that it could have. Now, there's some interesting characteristics that go along with that. Fats that are made from saturated fatty acids, I already said, tend to be solid. So they're called saturated fats if they're made from saturated fatty acids. And they tend to be solid at room temperature. So again, lard and butter, things like that, are saturated, and they tend to be solid at room temperature. And that's because they are packed more tightly together. Because those long fatty acid chains kind of lay flat, they tend to stack, and they can be pushed together quite a bit more, so they're more dense, and that means they're more solid at room temperature. The fats that are made from unsaturated fatty acids are called unsaturated fats, or a lot of times these are oils and liquids at room temperature. So most of your fats that come from plants and, for example, fish, they tend to be unsaturated. So these do not have the ability to pack closely enough together, so they're generally a liquid at room temperature. Now, if you know anything about health and diet, you know that if you eat too many of these saturated fats, that tends to cause heart disease, cardiovascular problems, and what we call plaque deposits within your arteries and veins. These saturated fats tend to clog up the pathways where your blood flows between your arteries and veins. There's something else you probably have heard of before is hydrogenation, or some people would pronounce it hydrogenation or hydrogenation. And that's where we take unsaturated fats and we chemically process them to add hydrogen to make them to where they are saturated in a way. So what we do is, for example, we'll take vegetable oils, which are unsaturated, and we would hydrogenate them. and that creates unsaturated fats that have double bonds that then can be packed more tightly together. So unlike the normal unsaturated fats that have the kink and that can't be packed closely together, now that we have hydrogenated them, they can be packed closely together. And these are called trans fats. And they actually oftentimes are considered worse than the normal saturated fats. So what we're doing is taking a normally unsaturated fat and we are artificially saturating it with hydrogen and creating some different molecular bonds. So why would we do this? I mean, if this material is even more unhealthy for us than your normal everyday animal fat, why would they do this? Well, it's a cheap way to get the consistency of more expensive animal fats. So you can take vegetable oil, which is far cheaper than, say, butter or lard, and you can hydrogenate it and you can make it into margarine. So it's got a more solid consistency. And it's also used to help prevent separation. Like in your peanut butter, no one likes to have their peanut butter where the oily part floats to the top. So if they hydrogenate those unsaturated fats within the peanut butter, it gives it a more solid consistency and it doesn't separate over time. If you ever buy like the all natural organic peanut butter, you may notice that tends to be a problem. If you don't mix the peanut butter up, you let it sit on the shelf for a while, the oils tend to rise to the top. So rising to the top, those oils would be unsaturated fats, and they become liquid generally, or they stay in a liquid form at room temperature. And then further down in the peanut butter, there's also some saturated fats that tend to stay solid. So hydrogenation is where we take those unsaturated fats and we chemically alter them and we artificially make them saturated fats, which doesn't seem like it would make a big difference, but it has a different effect on the body and can be quite unhealthy. So certain unsaturated fatty acids are not synthesized in the human body. The body has the ability to create some of these fatty acids that we need, but not all of them. So there are certain unsaturated fatty acids that our bodies cannot make and we must get from eating other materials. And one of these is called omega-3. Omega-3 fatty acids are required for normal growth and development, and they're also thought to have some sort of ability to provide protection against various types of heart disease, cardiovascular disease. So within your diet, you have to eat certain things that contain this omega-3 fatty acid, and some good examples would be some of your tree nuts like walnuts, also things like soybeans, spinach, fish. Those are all good examples. You may have even seen ads on TV where now you can get a pill that contains this omega-3 fatty acids. And they usually get that from soybeans or fish sources or something like that. So if your mom or dad take fish oil pills, that's part of what they're getting in that fish oil pill is this omega-3 fatty acid. So what's the major function of these fats? Well, in animals, which would include humans, the major function of these fats, these triglycerides, is energy storage. Remember, we talked about in plants how starch, which is a polysaccharide, is their main form of energy storage. And in animals, we also have a polysaccharide energy storage called glycogen. But remember, I said that was a very short-term type of storage. Well, the long-term storage for animals is fat. And fat is a good way to store energy because fat is twice as dense in energy as starch, which makes it handy for animals because animals have to carry around their energy source with them. They're not planted in one place like plants are. So we need our energy source to be as compacted or as dense as possible, and fat is what does that for us. Humans and other mammals store fat in what's called adipose cells or adipose tissue. And adipose tissue, of course, you're going to find in your lips and in your rear end and around your waist and throughout your abdominal cavity. And adipose tissue is primarily for energy storage, but it also acts as a cushion for vital organs. So if you open up a human being inside along with their heart and all their internal organs and digestive system, there's going to be adipose tissue throughout the body cavity. And that's not just humans, that's virtually all mammals and other animals. So it's also very important for cold weather and marine mammals like whales and seals because it provides not only protection, but it also provides like an insulation ability. So animals that live in very cold climates or that swim in the cold oceans, they tend to have a lot of this adipose tissue, not only for energy storage, but also for its thermal properties and for insulation and for protection of the inner organs. So we said there were three types of lipids. We said there were fats, phospholipids, and steroids. So now we're on to the second one, the phospholipids. The phospholipids are important because they are a major part of the cell membrane. So within your body you have cells that all have a cell membrane. These cell membranes are made up of these phospholipids. Two fatty acid chains and a phosphate group connected to the glycerol. So instead of three fatty acid chains, we only have two fatty acid chains and what's called a phosphate group that is attached to that glycerol molecule. And I'll show you a picture in just a second. The two fatty acid tails are hydrophobic, water-hating, or water-avoiding. But the phosphate group that's near what we would call like the head is hydrophilic, water-loving. So now we have a molecule that kind of has two separate ends. The long tail chains are hydrophobic, but the head part where the glycerol connects is actually hydrophilic. So here's a picture. We kind of have a structural formula and then a model right here. So there's the two fatty acid chains. Notice this one is kinked because it has a double bond right there. So that would be unsaturated right there, and that one would be saturated. So it has these two fatty acid tails, and these two tails are hydrophobic. They do not care for water or do not mix well with water. But then at the head, where it connects to the glycerol, there's a phosphate portion right here. And the head part is hydrophilic. In other words, it is attracted to water or mixes well with water. If you were to if you could possibly take a handful of these phospholipids and toss them into a container of water, they start to self-assemble in an interesting way. They kind of form this two-layered combination or construction, or this bilayer is what we call it. What happens is all of the hydrophobic tails, the fatty acid chain tails that don't like the water, end up pointing inward toward the interior, and then the phospholipid portion of the head tends to point toward the outside because it is attracted to the water or hydrophilic. So cell membranes are actually made up just like this. These phospholipids form a bilayer, and you can see they're kind of tail to tail. I'll show you a bigger picture in just a second. And this is what allows or keeps liquids in the cell and out of the cell. And this is what allows for control of what comes into the cell and out of the cell, this bilayer of phospholipids. So here's a closer up picture. So all the portions that are hydrophobic will point in, and all the hydrophilic heads form a layer on the outside. So it's called a bilayer, bi meaning two, because you have two different layers, these phospholipids, and they organize themselves in this way. This is what forms the major part of any cell membrane for any living creature. The last category of lipids we want to take a short look at is what's called steroids, and you've probably heard this before, having to do with bodybuilding or weightlifting or other things like that, or perhaps you've even had to have shots of steroids to control some inflammation or something like that when you were sick. So steroids are a kind of lipid, and they're an interesting kind of lipid because they have carbon skeleton rings. So instead of a long carbon chain, they have carbon rings, and it consists of four fused rings. And I'll show you a picture in just a second as well. And then off of these four fused rings, they'll have various chemical groups that kind of stick off like branches off of these four fused rings. And what really matters is not the four fused carbon rings, because that tends to be the same for all the steroids. It's what's sticking off of the edges. That's what makes the difference between them. Like, for example, the various vertebrate sex hormones are steroids. So you talk about testosterone or estrogen or progesterone. Those are all steroids, and the difference between them is kind of the branches that are sticking off of these four fused rings. Another steroid you've probably heard of is cholesterol, and that's an important part of cell membranes and the cell itself. And you have to have a certain amount of cholesterol in your diet. It's essential to animals. But if you have too much cholesterol, that tends to cause problems. It tends to cause clogging of the cardiovascular system, and that can lead to strokes and heart attacks and cardiovascular disease. So you've probably heard of people on a low cholesterol diet. Well, cholesterol is a lipid. It's a type of steroid, and your body has to have some of it, but some people get far too much in their diet. So let's take a quick look at a drawing. Here it is. Here are the rings that we were talking about, the 4 fused carbon rings, and then the different branches that stick off of these carbon rings determine what we're looking at. This one happens to be cholesterol. And if we looked at, say, one of the sex hormones, the steroids like testosterone or estrogen, it would have these same 4 fused carbon rings, but it would have different groups, functional groups, and other molecules sticking off on these branches. So there we go. Those are the lipids. We have the 3 that we talked about here. I'll go back to the beginning there. There we go, right there. So the lipids are fats, phospholipids, and steroids. They are fairly large biological molecules, but oftentimes they're not considered macromolecules or polymers because they're not quite as big and long chains and as complicated as some of the others. But they are an important part of living creatures and what living creatures need to survive. We'll stop right there and then we'll move on to 2 of the big ones, proteins, which is a really big one, and then the nucleic acids will finish us off